Thank you very much. Online and in-person guests who brave the elements and the crowd control security at the front. Good afternoon and good evening, good day to you. I've had a great pleasure to be here for a couple of days, and it's my privilege to discuss in the framework and the context of infrastructures, opportunities and challenges inherent to CRISPR in general and applications of genome editing in particular. So, as Dr. Olivier mentioned, I've worn different hats over the course of my career, some in industry, some in academia, some in academies, and then some in the real business world as an entrepreneur. And what I'm going to do is, for maybe 30 minutes, walk you through the business that we're in collectively. Whether you're a true academic, a true entrepreneur, politician, scientist, a political scientist, or any combination thereof, what we often try to do is to turn our deep knowledge of science into technologies. CRISPR is a good example of how our knowledge of CRISPR immune systems and bacteria has been translated into a genome editing tech, Nobel-worthy genome editing technology, which sometimes can be applied to generate products that hopefully will change the world. Change the world for patients, change the world for individuals and their health, change the world for agriculture, and change the world for infrastructures, as I will argue today. And then, of course, in today's scientific skepticism age, even here in Europe, where vaccines were first developed, right, by Pasteur, just down the street, to some extent, Pasteur, acceptance of science and technology is a big challenge. And as a matter of fact, bridging different topics of science and technology, political science and acceptance, science communication, science advocacy, science governance is extremely important, and acceptance is something I will finish with today. So let me give you a few quantitative insights to understand where we are and where we're going, and why super forecasting what will happen in the next 10 years, the next 20 years, the next 30 years. I'm actually going to talk through genome editing in 2050 today. That's the ambitious goal that I have. Let me tell you where we are now to get a sense of how fast and how much things are changing. Because this is arguably once in a lifetime technology, right? So if you look on the top left here, about 250,000 labs in the world have received a CRISPR construct. Of course, anytime.
Are those gene data or?
Those are gene data.
Okay.
Yeah, data from gene that's published. Right. So those are gene data. So 250,000 shipments in the world already. And those shipments have been distributed to 104 different countries. So imagine making a list. I know we have some presidents downstairs from South America, as a matter of fact. 104 countries have access to that technology, not for profit at this point in time. Now, what's interesting is it's very, very competitive. There are geopolitical forces that are driving competition, notably between the US and China, and to some extent, even Europe. And if you look at the data today, right now, almost 60% of all the authors in the world who publish on CRISPR are residents of the United States. Number two is China, and China is growing fast. I'll get back to this in a second. And ironically enough, half of the top 10 hail from Europe, 4 in the EU and the UK. And it's interesting given the regulatory challenges inherent to the exploitation and deployment of CRISPR-based genome editing in this particular geographical location. In the bar in the middle, if you really look at the pace of growth in China, there are reasons to be aware of it and perhaps somewhat concerned. I'll get back to that in just a minute. And Europe is not growing as much as it should. Maybe predictably, maybe disappointingly, or maybe there's opportunities for investments by governments and banks and others. And what's interesting in the top right is that 95% of all that work is focused on human cells. Another 4% of their work is focused on animal models of human health.
How broad is your human cell definition? Because with the cell lines,
any human cell line.
Yeah, so are they
any human cell line
at that point?
Well, they're humanized.
Yeah.
They're imperfect proxies of human health. Yes, they are. That means that 99% of all that work is focused on human applications. which is great if you care about human health, which is tragic because it means that everything else outside of humans is less than 1%. I can get back to that conundrum in just a minute. And then, of course, there's all kinds of things you can do with CRISPR. And I'm going to talk about all the different applications scientifically that are there. Now, since we're at the Sciences Po, I'm not going to just look at this as a scientist. I'm going to look at this as an observer. What does that actually mean? The same data seen a different way. Well, it means that it's democratized. This technology is democratized across the world, and the average lab can use CRISPR. As a matter of fact, in North Carolina, people who are in high school learn how to use CRISPR in the lab. My own children have taken a CRISPR class in North Carolina as teenagers, and they've had CRISPR homework and they've had CRISPR experiments. I can help with that. That's the pace at which it's changing. There's dozens of CRISPR classes at NC State. At all levels of education, you can take CRISPR class, fundamentally, philosophically, communication, politics, policies. You can also take manipulation of organisms with CRISPR in a classroom and edit and manipulate and genetically engineer an organism. And then you can use that knowledge to do that in the lab. There's probably hundreds of labs at NC State that use CRISPR. So it is democratized. This is a very global market. And investors, whether they're banks, for-profit, not-for-profit, governments, employees, employers, inventors, licensors and licensees, there's a whole CRISPR business I'll talk about in a few minutes. It is very networked. I think this is a theme that came up in yesterday's conversation to some extent. Not any one group, not any one institution, not any one individual, not any one university, not any one company can do it alone. And transdisciplinarity and interdisciplinarity and strategic collaboration within academia, between academics and industry, between industries, and public-private partnerships are very important to really deploy at speed and at scale towards building infrastructures that will be relevant in 2050. I'm going to make that argument today. Since we have some bankers online, let me show you what's at the bottom here. Those are 5 different segments of CRISPR-based startup companies that have been sprouted in the last 10 years. There's about 50 of them. And I'm not talking about startup companies that use CRISPR, but there's thousands of those. I'm talking about startup companies that were founded based on CRISPR technology. The ones on the left do tools, the ones on the top, first generation therapeutics, the ones in the middle here, do agriculture, talk about that. The one at the bottom is microbiome, the one on the right is second generation therapeutics. This is where bankers and venture capitalists invest their money. That's good for human health, but there's not enough investments going everywhere else. I'll discuss that. That means that it's very focused on the top right, but there's huge opportunities for agriculture, agricultural ecosystems, which I'll discuss. And then obviously there's rising competition in China. If you look at the metrics, the patents, the investments, the institutes, the recruitments, the patents, and the VCs, the extent of governmental level policy to invest on CRISPR-based agriculture in China is very real, very tangible, and very concerning. So this is not a scientific lecture, so I'm not going to talk about CRISPR science too much. If you want to learn about it, you can invite me back, and I'll talk about all the idiosyncrasies and the beautiful genetics of CRISPR in bacteria. But the reason I was invited is not to talk about this, to talk about this, to talk about human. So let's just go straight to technology. CRISPR science is great. There's many lectures, many papers. But the reason CRISPR is famous is because CRISPR can be co-opted as a genome editing molecular tool. So in the audience here and online, I'm sure you've all used a razor at some point in your life to shave a part of your body I don't need to hear about right now. And what CRISPR is in nature is a molecular scalpel that can be used precisely and programmably to cut DNA, like a razor. And what it can do is imagine a stack of books in a library at Sciences Po, right? With maybe like 3 billion letters in it. And you can use CRISPR as Control F and look for Sir William Shakespeare's famous quote, "To be or not to be, that is the question." And instantaneously, CRISPR will find that sequence in the book of life And it can be programmed to cut precisely between that and it. And at that location of cleavage, you can program CRISPR to edit the DNA. To be or not to be, hashtag the question. If you like Twitter, though Twitter is having a rough week. You can insert something, if you're philosophical, it's also to be or not to be the quintessential question that defines humankind in social sciences. Or to be or not to be, period. You can add DNA, remove DNA, change DNA, rewrite DNA. This is what CRISPR actually is as a technology. As a matter of fact, this technology has been used and deployed in what I call the CRISPR zoo, and they're related to ecosystems. Those 250,000 labs and 60,000 published authors that have published 35,000 papers as of this morning, US time, have implemented and deployed CRISPR across the tree of life. The power of genome editing. So at the bottom right here, you see model organisms we use in academia, viruses, bacteria, yeast, all the way to non-humanoid chimps, also used for human health manipulation, and then model plants like Arabidopsis. In the real world of agricultural ecosystems of high importance to the European bioeconomy, CRISPR crops, CRISPR vegetables, CRISPR fruits, CRISPR livestock. So think of a CRISPR mushroom, CRISPR tomatoes, non-browning mushroom. We can knock out a gene in mushrooms responsible for the browning and the shorter lifespan of a sliced mushroom available at the supermarket. Non-browning white butter mushroom from Penske. Or we can think of a leaner chicken with higher portion of meat compared to fat, or less fat in bacon from pigs, or a more healthy tomato, or should I say tomato, in the UK market already, and on sale in Japan for 6 months already.
I have a question on the pig. So recently we saw this troubling story of the patient that was given a modified nuclease genome edited pig heart. So turns out there was a big debate because this person had stabbed someone that has then got the heart, then died because of apparently a herb, so a porcine virus.
Correct.
That infected him. But I never got, and journalists I asked who are well, aware of this field, asked whether it was CRISPR or the fingers or talents that enabled the company to produce such art. So do you have your own opinion on this? Do you think it's CRISPR? Do you think it's another?
So I don't know the definite answer to that. I'm not sure it's a CRISPR nuclease.
Okay.
I think it was the first generation already underway. If you look at the people involved in the timelines, circumstantially, more likely than not, it wasn't CRISPR.
Okay.
But as a matter of fact, George Church
It's trying to do that.
had already worked on addressing PERS by reprogramming multiplexibly CRISPR to remove retrovirus.
And so it's working.
It is very promising. I don't think it's working in vivo as of yet.
Okay.
And then, of course, at the very top of social sciences, at the very apex of the tree of life, human navigation and medicine. I'll get back to that in just a minute. Now, what's interesting to know and important to know, even though I'm not going to talk too much about the science, is that those 250,000 labs and 60,000 authors, 34,000 papers published to date have been very hard at work at doing what I'm going to call sharpening the CRISPR toolbox. And as a matter of fact, my lab and hundreds of other labs have advanced and matured and sharpened and expanded the CRISPR toolbox. So virtually we're able right now to edit and manipulate virtually the genome, the transcriptome, even the epigenome. We can reprogram epigenetic imprinting from mother to offspring, or in patients, or in people, and or in animals, and or in livestock and plants today. We can virtually reprogram at speed and at scale the genome, transcriptome, or epigenome of any species on planet Earth. That's where we are right now. So CRISPR is a 10-year-old technology. I think some of you have seen a 10-year-old before, right? It is the most promising 10-year-old we've seen in the last few decades, maybe since PCR, as a matter of fact. One, you can read, programmable, specific, transferable, efficient, precise, portable, reproducible, scalable. It's an amazing molecular tool that high schoolers in North Carolina can use to edit genome. But it's imperfect as a 10-year-old. Think about the cell phone, the first cell phone 10 years in. It took a lot more than 10 years to go from what it used to look like to where it is now. Or think about Minitel to internet. It took a long time. Think about, the first projector, your first computer, machine learning 10 years ago versus today. Right? CRISPR 10 years in is already in the clinic. But it is imperfect. It's a little fat, 10-year-old, throws a tantrum here and there's not always predictably efficient. Throws a tantrum. But it's good enough for the FDA, bottom line. It's good enough, safe enough, efficacious enough, and I'll get to that in a minute, okay? That convincingly it can be deployed at speed and at scale. And there's a lot of efforts right now going on to understand how we can enhance DNA repair, and we can deliver CRISPR more efficiently. And the big question that will shape the next few decades to build infrastructures, this is where the investments need to go, is what organisms to edit. And what for? That's the big question. Now that we can do it, what ecosystems can benefit the most from CRISPR-fueled genome editing? That's the argument I'm going to make today. So what are the applications? Well, just in my lab, just as a microcosm, pun intended, as a microcosm of what can be done, one group, one lab, one location, one team of about 15 people. We've shown in the last 10 years, we can use CRISPR in bacteria to kill viruses. We can use CRISPR in bacteria to edit viruses. We can use CRISPR in viruses to kill bacteria. We can use CRISPR in viruses to edit bacteria. And as a matter of fact, just today, a paper came out showing we can use CRISPR in bacteria to edit a virus with another CRISPR to edit another bacteria. For those of you trying to keep track online. So one team, one location, one lab can do all those things and develop novel technologies that enable us to do that at scale. And there's hundreds of labs like mine. And what we do collectively is we try to understand what particular elements constitute the tools that we can use and deploy for CRISPR. Once we understand the science behind CRISPR, then we can go into technologies to type bacteria, track bacteria, immunize them against viruses, do the transcriptional control, genetic control, epigenetic control, tag organisms of interest, kill organisms of interest, and or screen for natural variants in nature that we can use for breeding. And what have we done already? Well, we can work in the farm land and infrastructures. We can work in manufacturing, bioproducts, food, ag, biotech, biofuels. We can work in the food supply chain. As a matter of fact, if you've consumed yogurt or cheese anywhere in the world in the last 10 years, I can guarantee you've consumed a product that was fermented using a CRISPR-enhanced phage-resistant dairy bacteria, including developed in France, between Tours and Poitiers, in labs of some of the best developers in the world for startup culture. And then, of course, human engineering, not for humans, for the microbiomes that we exist with. And I'll get back to that in a minute. So that's theory. That's theory. It's theoretical. It's an academic exercise. But who funds our work and what do we do with it? Well, I went to business school. I got an MBA and I like the real world. And it's easier to get funding from companies that know what they want than governments that are not sure what they want to do with it. So my lab, in just 10 years, is working with all those people. So some big European people like DuPont, like Novozymes, like BASF, right? Some large companies like Syngenta, like Corteva, like Intelia, and then some startup companies like Locus and Trico and CRISPR Biotech and others. One lab, one location, one team. And all 16 people who got PhDs and masters from my own lab all went to industry to work with those companies. Why? Because they can change the world in real time. As a matter of fact, What kind of ecosystems are we talking about? I'm going to give you just a few examples because it's not about science today. I want to just contextualize the infrastructure conversation. So, for example, a paper that came out yesterday from my lab, I can't make that up, is showing how we can use CRISPR bacteria to control the soil microbiome, and by extension, control plant growth and plant health. Like, we understand our microbiome in our gut is important for our digestion. We can change and manipulate the microbiome in soil to enhance plant growth and plant health. Think of climate change and sustainability and an affordable food supply chain. I'll get back to that. Likewise, we can do that in food, but dairy cultures, cheese, yogurt, and anything that's fermented. We're not doing champagne yet. There's not great champagne in the US because we don't have champagne, right? It's bubbly wine. It's not quite the same. Same with wine, right? Fermented dairy products, fermented vegetables. Think of the market for kimchi globally, or sauerkraut in Germany, or pickles in France, cornichon, right? Of course, we do that in animals, enhancing animal health at a time when antibiotic resistance is a huge issue. So can we make the microbiome of livestock more healthy, like humans? And then, of course, all human microbiomes, oral health, gut health, vaginal health in women's health, and skin health for beauty or aging, for those who don't want to change their own DNA to do that. One lab, one location, one set of examples. So let's just zoom out here and look at the bigger picture. 104 countries, 250,000 labs, 60,000 published authors, 35,000 papers. What are they working on? Well, they're working on tools, guides, enzymes, software, plasmid delivery, kits, primary cell lines used to research and understand the biosphere, or parts of the biosphere, as we heard yesterday in Dr. Libria's presentation. Pieces of the biosphere, a piece of cheese, a piece of yogurt, a piece of a farm, a piece of a territory, a piece of soil, or a whole field. I'll get back to that. In biotechnology, any bacterium, any yeast, any algae used in food, manufacturing, household care, or bioenergy has had its genome enhanced industrially already. We published some of that in 2017, 5 years ago, almost 6 years ago, in a month, 3 weeks, and 17 days. That's the pace at which it's changing. How's the biofuel need in Europe right now, at a time where energy constraints are significant? The cost of the price of energy is significant, and availability and affordability of gas is a challenge that I understand, though I'm just an American, right? And then, of course, agriculture. I went to an ag school. I'm an ag guy. I used to be French, I can relate. Right, so plants and crops, obviously animals and livestock, obviously microbes, as just discussed. I'm going to talk about forestry for the context of infrastructures, because that's where we're going in 2050. Flowers, think of Holland and the Netherlands here, ornamentals, and of course, aquaculture, when the protein distribution to feed the world is going away from livestock more towards plant-based proteins and fish. Check, 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 check. And of course, humans. There's so many things you can do with CRISPR, gene therapies, antivirals, infectious disease, microbiome, antimicrobial cell therapies, immunotherapies. We've already heard about xenotransplantations with human hearts, gene drives, invasive species, of course, diagnostics, and even animal care and pet care. Okay, so since we have some bankers online, and since I've been in the business of CRISPR for 19 years, which makes me a dinosaur. This is the infrastructure that investors are building. They're building next generation pharmaceutical companies.
So this is mostly private investors, right?
Private investors, that's where I'm going. So right now, it's only being built by private investors, primarily. Private investors are short-term focused. They don't look at timelines that extend past the next 20 years. More often than not, they don't have even NPV models that enable them to invest past the next 20 years, let alone by the next 25 years, certainly not 2050 with climate change. And what are they focusing on? They're focusing on therapeutics. Why? Because it's tangible, there's a pipeline, there's milestones, it needs to be very lucrative. So what are people doing? Well, they're working on CRISPR therapeutics. And what are we doing? Well, saving human lives. That's great. It's very important. I actually do that. I have multiple companies that do that. But how many lives can we really change? And how many patients can we dose in the next 10 years? According to my own estimates, aggressively, maybe 100,000 people.
That's not bad.
It's not bad. It's 100,000 people. 100,000 people. Up to 100,000 lives in patients that may get dosed and benefit from a CRISPR therapy. It's amazing.
Ex vivo or in vivo?
Either.
Either. You think you're going to have
Either.
the next decade an improved in vivo?
Oh yes, I will take that money. Yeah, I'll place my own money on that. I've placed my own money on that. I'm convinced.
Despite delivery and.
Despite delivery, absolutely.
Okay.
I'll get to that again in 2 minutes.
All right.
I believe in this. Now, what's the number if we do that in ag? What's the number, you think? How many people can we feed if we strategically invest for CRISPR in crops and maybe livestock? The next 10 years.
Well, but then your question implies that we don't already feed them, but we do,
To
right?
some extent.
Well, to a large extent, like people dying of hunger today are dying of hunger because of wars and conflicts,
Correct.
right? So, I mean, okay, so I guess your question is more like how many people do we think will benefit from CRISPR modified crops
In the next 10
in
years.
the next decade?
Who a number?
Well, if you take the US, I guess you can already would have 1 billion. people.
Yeah, I mean, I think it's in excess of 100 million people.
Oh, okay.
Maybe the cap is 1 billion, maybe.
Yeah.
10 years. So we'll talk about that in a minute. How about forestry? If we say we're going to use CRISPR and invest strategically into infrastructures that will enable us to breed a more sustainable forest, how many people on planet Earth could benefit from that?
Probably future benefit right there.
So how many people could benefit from a more effective carbon capture? How many people
Did
could
you
benefit?
really capture carbon when you grow trees? Because then, okay, it so happens that that Friday, I was in a conference on precisely this.
Yeah,
And one of the people were arguing that carbon capture by planting trees is not really carbon capture because then the tree dies. And then what do you do with
well, it
the tree?
depends what the lifetime of the tree is, what the lifespan of the tree
Yeah.
is, what the use of the tree is. Not necessarily. Sometimes it lives hundreds of years.
Okay. So
Sometimes
you're
it lives
planting on
tens of years.
Okay, so secular tree.
Yeah.
Okay.
So how many people could benefit from that? I think it's more than 1 billion. But we'll get back to that. Okay. So hang on to that thought, because we're trying to predict the future, but let's just look at the present. So already in the US, there's some people who could be giving a talk, people who are on the radio, NPR, people who are on TV in the news, who can walk around and say, I had my genome edited. I mean, I didn't edit my genome personally yet. But people have had their genome edited. Dozens of patients, a few dozen patients. It's happened. There's people deambulating on planet Earth today who can say they benefited from a gene therapy, some ex vivo, some even in vivo, not so far from here in the UK, who have had a genome edited. And they survived and they benefited tremendously from that. As a matter of fact, there's 48 different clinical trials documented. Clinical trials that go all over the world, primarily US, Europe, China, there's one in Australia. And there's 24 that are recruiting actively right now as we speak today.
How about the people who pulled the Josie Alzheimer's stunt? That is injecting themselves with Get9. Do you count them in?
I don't count it in. These are clinical trials.gov. And
No, but so on one more, I guess, technical level.
yeah,
Do you think that without a potent vector, Cas9 somehow persist or then
No.
get
Short-lived. Short-lived, managed by the immune system,
Okay.
destroyed short-lived. Yes. And I think I would say that it's a form of natural selection to some extent, with all due respect to Josiah and others. I mean, developing your own drug is not a good idea, generally speaking, especially in today's medicine world. And the New England Journal of Medicine has published multiple examples just in the last year of world-class clinical trials showing it can be done in vivo in patients. Beta thalassemia, sickle cell, leukocytosis, and ATTR, including in the UK. This is just the beginning of the future of medicine. But if you want to build infrastructure, you're not going to build more people. We have plenty of those already, and as a matter of fact, it could be a problem to have many. So what does the World Economic Forum say, since we have bankers on the online? Well, the World Economic Forum, not me, is projecting that by 2030, 100 million farms in the world will plant a genome-edited seed. We're 7 years away. 1 month and 13 days, like that. That's a lot of farms. 100 million farms. I don't know how many will be in France. I don't know how many will be in the US.
Okay, so I have a question on this because I met the person who I think wrote the smooth numbers from the WEF. And I guess that, yeah, so those projections and those views are used by, of course, an attempt to lobby governments. But it happens that in the US, we saw recently the creation of a new agency under the NIH, and this person, her name is Sonia Bergen, she used to work for DARPA, and she funded the CRISPR project, and she went to Ginkgo, now she's heading this brand new agency. Do you think, so it's NIH, it's not really ag, but arguably could to some extent be, it's a whole question of what is, I mean, between the USDA and the FDA, and what is a drug, what is a plant, this food, whatever. But do you think that it shows that this kind of projections and lobbying and narratives are currently really aggressively working in the US, or do you think it's not being really triggered yet?
Oh, it's been triggered. It's happening.
Okay.
I know running virtually.
Okay.
And she was on the advisory board for Jennifer's Innovative Genomics Institute.
Yeah. Okay.
And we know, and actually I hosted a meeting at the National Academy of Sciences in the US just last month, bringing together FDA and USDA and EPA to talk about regulatory frameworks for genome editing in the US moving forward. And in FDA, they're very D-centric drugs, but FDA also does food. It's the Food and Drug, not Food and Drug Administration. So absolutely have confidence that NIH investments towards understanding how the agricultural infrastructure is critical for health and disease management in the US and sport is very much part of the picture.
Okay, so follow-up question.
And it's cheaper.
Yeah.
It's cheaper to build a good, sustainable food supply chain than it is to therapeutically dose patients.
Okay. And so do you think this will, so there was a question of whether mammals, animals would be FDA or USDA. I think last time we heard it went from FDA to USDA. Do you think, do you know how it's going to evolve, or do you say like this?
Yeah, I mean, I think it depends how the animals are being used and how they're being controlled, how they're being disseminated, right? So they're used for tissues and transplantations, and clearly that's FDA. But in the end, the grading of beef is USDA grading.
Okay.
The role of the US Department of Agriculture is to manage the US Department of Agriculture. That's where they're going. I'll go back to that in a minute. So can we build a resilient, sustainable, healthier food supply chain through innovation and technology? The answer is yes. As a matter of fact, we know about not just the scientific news, but the public news communicating about CRISPR. The cover of the New York Times Magazine, Wired, and of course, G. So we've heard about livestock, dairy cultures, the non-browning mushroom, here we go yet again, tomatoes, corn, rice, and others. And if you scope that out and you look at China, and you scope that out and look at investments in Southeast Asia to secure their own supply chain with land they manage from across their borders and seeds they're developing in-house using the ChemChina purchase of Syngenta and others strategically. We know where things are going. But let's talk about forestry for a few minutes. This is how we're going to build infrastructure. I'm convinced. I would make the argument. I know because I'm doing that. So I think to me, the future of humankind is partially dependent upon addressing climate change. And we heard about carbon capture. So let's look at carbon distribution on planet Earth. Right here, on the top left quadrant, is the carbon distribution as it currently stands, that's sequestered on planet Earth. And if you zoom on the annual part here, right here is humans, 0.06 gigatons. We're not very significant. We're not a good means to capture carbon, evidently. Nor are animals of livestock, as a matter of fact. Fish much better, 10x, right? But the large majority of all the carbon on planet Earth is captured in living entities that are plants. And of that, 70% is trees. 70% of carbon on planet Earth can be captured in trees. So where should we start? I'm saying we should start with trees. Why? Well, because fiber and timber construction, climate change capture more, bioenergy and bioproducts, and of course, emerging markets as alternative plastics and other petrochemical related industries is very defensible, justifiable, timely, and relevant. The challenge for investors, to go back to the bankers on the call, is they don't have the luxury or the appetite for this kind of timeline, those kinds of investments. Only governments and very long-term investors have the capacity and willingness to invest in genome editing to create a better forest. But it's hard to scale up and it's hard to deploy. Well, how hard is it? Well, let me tell you, I know because I've been working on this for years. The same concepts that we use for human genome editing in the clinic, the same concepts we use for human microbiome editing in food, the same concept we use for genome editing of plants and crops can be used in trees. But it's harder, it's slower, it's longer, and it's more difficult. I'll make the argument, we'll see if it goes in the Q&A. I'll make the argument that it's 1,000,000 times harder to edit a tree than a human. We can discuss that at your curious. Right? Based on the size of the genome, the availability of people, the knowledge that we have, the protocols, the timelines, and the work inherent to it.
Do all the trees have the same employee? How many birds?
They do not.
So the ones you're working on, how many?
Up to 5. Some of those genomes are 10 times bigger than the human genome. 30 gigs. Not 3 billion letters, but 30 billion letters in one genome. It's harder, much harder. And we know a lot less about tree genomes than we do human genomes, trust me, sadly. Right, but it can't be done. You know how much we know about tree stem cells? How easy it is to regenerate tree stem cells?
Say that the trees are 10 billion.
30 billion. Some trees are 30 billion. We're 3 billion.
Yeah, 10 times.
Some trees are 10 times bigger. Yes. Wow. But they live a lot longer and they can't move around. So they need to be able to do everything on one location. They're able to do photosynthesis. We can't. They can't do many things we can't do. They have much more stress, biotic stress, tougher lives. They live so much longer, hopefully, than we do. Depends on the people, depends on the tree. Different philosophical conversation here, but absolutely, yes, 30 billion letters. Can it be done? Well, of course. I do know because I've done it. It's not trivial. So there's 20 years of work from my favorite collaborator, Jack Wang, who's one of the best tree geneticists in the world. He spent his whole life doing tree genetics, and he's developed the most sophisticated machine learning based model of tree genetics in the world. And what did he do? Well, we look at traits of interest, design CRISPR constructs, transform tree stem cells, harvest edited trees, confirm that they are what we think they are, check the phenotype, and iterate that process.
And so the third step to fourth step, like this iteration, how long does it last?
Well, that's the next slide. Just hang on to your thought for 1 minute. The data tells us that if we want to grow trees that have the same growth pattern, the same height, same weight, the same diameter, the same density, no drag, no cost, and all we want to do, quote unquote, is manipulate the lignin composition to make them more efficient and sustainable. There are 69,000 genome editing strategies we could use in the lab. 69,000 and a few. Of those strategies, the machine tells us confidently only 347 will work. What does that mean? It means that somebody who doesn't have the tree genetics knowledge like we do will waste 99.5% of their time trying to edit trees. That's why we do human genetics, because we know the one gene associated with the one disease. But in trees, we don't. 99.5% of the time, they're going to be wasting their time and money. Usually it's their time, somebody else's money, but we'll get to that. And tragically, more often than not, we need at least 6 genes to be edited. That's hard. It's 6 times harder than changing 1 gene. And if it's polyploidy, it hurts my head. But we can do it. So how long does it take? Well, so to do a lab in greenhouse proof of concept is 2 to 4 years. To do a field trial is 4 to 8 years, to 4 to 6 years. And commercial scale up is 8 to 16. So what we're working on for 4 years, if everything goes right, will be commercialized in 2046. Let me say that again in case you missed it. So we've been working on this for 4 years. I spent 4 years of my life working on genome edited freaking tree.
Will it work?
Of course it works. Next slide. 1 second. It's hard. It's slow. It's painful. And if we do everything right, the world will benefit from it in 2046. I don't want to know how, I know you don't want to know how old I'm going to be in 2046. I don't want to know how old you're going to be in 2046. When I say that, and I talk about this to investors, they don't want to know what the world's going to be like in 2046. But if we want to build infrastructures, we need to have the foresight to invest. Because if we miss 1 or 2 steps, it's going to be 2050. And if we don't make it by 2050, what's going to happen to the planet? We know. So, can it be done? Well, of course it can, else I wouldn't be here talking about it. We have edited trees in the lab in the greenhouse. Not quite as tall as the ceiling, but as tall as this beam. Slightly taller than the beam, as a matter of fact. Slightly taller than the beam. A couple hundred trees, hundreds of trees, but only hundreds of trees. Right? They have maintained their height. They have lower lignin, and we can change things like the color of wood. Why? So we can prove to people that the trees are edited.
Okay.
Right? It doesn't really serve a place. It's a flex. It's a big flex. And you know what? It took us 4 years to make 3 sheets of paper. But we can make CRISPR-edited paper.
Just a question, how expensive is it to maintain the trees? That is it very expensive?
And not too much. No, it's no cost. Much harder to do the genetics.
Okay.
Yeah. So what does that mean? As we build infrastructures, think of the scales. We can do a virus, a bacteria, a plant, a human, or a tree. We can do a single edit. We can do a large genome, 30 billion bases. We can apply it in something that's microscopic, 1 dose right? One dose. Or we can have to plant 100,000 hectares. 100,000 hectares of the forest. What is it going to take to do that? And then some of those organisms will live 20 minutes. Maybe a bacterium replicating in ideal condition. Maybe it will die in 20 minutes if you wanted to. Some of those trees we hope will live hundreds of years to capture carbon through the next century, not the next millennium. This is 1 century at a time. Let's build the forest, a large infrastructure, 100,000 hectares at a time. That will allow us to capture more efficiently, more safely, more responsibly, more carbon. Can we do that? It'll be more resilient to pest and disease and drought and the like. Well, if we can do it all, the challenge is the politician, the public. With great possibilities come great responsibilities. So the ethics of genome editing. Are we going too fast or too slow? How about all those moratorium and reports from the academies and the learned societies? How about public relations? I mean, if people don't want to eat CRISPR corn, are they going to hug CRISPR trees? I hope that's why they didn't come in today. No. You can't come in, right? Whose stories? And how are we going to regulate that? In today's USDA, CFR 7340. New regulation. What are the benefits? What are the risks? And how are we going to govern the ungovernable? It's a good question. Because we can't govern, or can we? We can discuss that all day. And if I try to foresight and forecast what's going to happen in the future, well, of course, we're going to work in the clinic. Of course, people will be more excited about this. Of course, Europeans will catch up in time. But by 2030, we won't be able to feed the world and address climate change the way we need to. As a matter of fact, by 2050, I'm not even sure we're going to be able to do that. Notwithstanding everything I just said and went through, it's going to be hard, right? And the new White House executive management, figuring out governance, figuring out collaborations, figuring out investments and how to build and fund this research is not trivial whatsoever. And all those things have been well chronicled in the aforementioned CRISPR journal. It's hard, it's difficult, it's challenging, it's not for everyone. And there's technical hurdles, scientific hurdles, commercial hurdles, investment hurdles, ethical hurdles, practical hurdles, governmental hurdles, public perception hurdles, on and on and on and on and on. And we have our work cut out for us. But places like this, I think, are well positioned to inform the public, to inform the governments, to inform the investors, to inform industry, to inform the public, to inform academics. We need more people who graduate with PhDs understanding CRISPR and genome editing to help inform policies moving forward. So the 30-minute conversation turned into a few Q&As already. I think this is just the beginning of the next chapter of how we can use genome editing technologies to build farm and forestry infrastructures that will help us not just to address human disease, but to feed a fastly, rapidly growing, uncertain, volatile world. And then also hopefully to address grand challenges like climate change at a time where it may very well be the biggest liability that we have before us. So thank you again for the invitation, and we'll just take questions.